Well, first, I'd like to express our appreciation to welcome Professor Eisenberg. When I was at UCSF, and we heard about your name, and I come to UCLA a number of times, but never had a chance reading you because I work in different field.
That was my loss.
And now it's a special honor to meet you and welcome you in Vietnam. And like you said, we look forward to the future, actually working together with the two very important schools. So with that, I think my questions or some sort of ideas about this is based on my experience in the US as well as here. When I returned to Vietnam, what I'm very encouraging about many important developments which you already observed, particularly with the stem cell lab. But however, there's one thing that I find is that we have a lot of need and pressure not to study the mechanism, not to study too much of the detail of some topics, but how to apply those knowledge, skill, and techniques into application, and to put it simple, into product. For example, some diagnostic product, applying nanotechnology, antigen epitope, or some of the therapeutic in terms of monoclonal antibodies, targeting therapies, which could express. So with that idea, I hope that in this discussion, we can come up with some agreement and some line of work in the future that we can have that achievement in Vietnam. And of course, when we send our students there, we like to have academic knowledge. They can lead the basic research here, but we do not forget how to apply it to the production in Vietnam. Thank you.
I'd like to start by replying to that most important question. First, historical illustration that I'm sure you're familiar with. Michael Faraday in 1851 showed that by spinning a coil of wire inside a magnet, he could generate an electric current. When he showed this to the Prime Minister of England, the Prime Minister said, well, what good is this application? And Faraday said two things. The first was, what good is a new baby? And then he said, Mr. Prime Minister, I'm not sure what good it is, but I'm certain that in time you will tax it. I have to say, in my own work, I started as a structural and computational biologist and became interested, as I will show tomorrow, in the amyloid state of proteins, something that you might think would be very far from applications. We then learned that one of the central proteins in cancer, p53, involved in at least 50% of all cancers, that forms an amyloid in cells. We went on to study that amyloid and to design an inhibitor. We've now shown that this inhibitor can go into cells, and in mice it can shrink tumors of ovarian cancers. Now, I had no idea, even 3 years ago, that we would ever have any application that might have some eventual therapeutic use in cancer. First of all, so I think this illustrates in a small way, as Faraday's work in a big way, that work in the fundamental area can affect other areas in very unexpected ways. So I think if VNU is going to be successful in the long run, it must emphasize fundamentals along with applications. And of course, you need both operating side by side. We could not have done our experiments in mice without my colleagues who know how to work with mice and to introduce our inhibitor into mice. So those were collaborations within the university. I'm very fortunate to have enjoyed my career at UCLA, where we have a medical school right next to the basic sciences, within 100 meters, so that we meet each other all the time. When we learned that p53 forms an amyloid, and we wanted to find cancer cells to see if p53 amyloid was in those cells, within 2 days we found colleagues that we had not met before in the UCLA Medical School who worked with ovarian cancer and prostate cancer and melanoma, and we found in all of those that the p53 amyloid, at least in some of the cancer cells. We also are fortunate in having a School of Engineering right across the courtyard, and with all these activities going on, the sort of collaborations are possible that lead from basic science to applications. I see the two going hand in hand, but that's a wonderful question that you brought up.
Because I come from Faculty of Chemistry, so I would like to have a question to you. Because in this program, you support for us to train the student in chemistry field. So I want to know what do you expect from chemistry field? I mean, you just consider in the basic chemistry, or the chemistry field can orient to biology or medicine? Yes, thank you.
Merkel was a chemist before she became Premier of Germany. Margaret Thatcher was a chemist before she became Prime Minister of England. So it seems to work in politics, but more directly in fields of biology and medicine. At UCLA, we have in our Department of Chemistry, we have medicinal and synthetic chemists who make new molecules, which then can be tested on cells through collaboration with our medical school. That has led to one of the most important new cancer inhibitors, which is now starting to support our Department of Chemistry in an important way. We have physical chemists, well, Professor Liu, who's a mass spectroscopist, will be speaking at this symposium, and these methods can turn up all sorts of new insights. So I think training in fundamental chemistry is a wonderful road to applications in biology and medicine. We don't know how the discoveries in chemistry will immediately affect biology and medicine, but important discoveries almost always do. We have to be alert to the wider implications of our work. I found a most interesting case, which I'll just take a minute to tell you about, which I'm going to write an article about whenever I have time. This is a young woman who was at Yale in 1955. She was getting an MD degree. At that time, Yale Medical School required a thesis. She did an experiment which she and her supervisor understood the experiment, but they did not think it was worth publishing. They never even discussed publication. 2 years later, exactly the same experiment was done by Chris Anfinson at NIH. He published the results of that experiment. Actually, his experiment was not as good as hers. It had to do with the denaturation and renaturation of ribonuclease. His experiment was published, recognized as being important in showing that the information in a protein sequence determine its three-dimensional structure. Several years later, he received the Nobel Prize in Chemistry for that work. I wanted to understand why they had not published their paper. So I went and talked to the supervisor and the student. I thought it was most interesting that the student was not in the slightest bitter. She said, I received a wonderful education from my supervisor. He was a wonderful supervisor, even though he did not think of publishing the experiment. And I asked her, how did you feel when Anfinsen published his paper? And she said, oh, I felt wonderful because my result was better than his. I got 40% recovery of activity. He only got 20%. I said, how did you feel when his result won all sorts of prizes? She said, well, that was only right. He understood the meaning of it. I did not. So what I learned from that is when we do an experiment, we always have to place it in the wider context as chemists, biochemists, cell biologists. And this is very hard to do. So we have to think about what our result means. Like she had a very simple result that she could renature a protein. What were the implications of that? For some reason, they did not place that in the wider context, but Anfinsen's did. All of us who do experiments have to be alert. What does my experiment mean? And you have to be willing to say, there's a new understanding here, a new paradigm, and take the boldness of writing a paper and pointing that out. I try and teach that to my students. When my students do an experiment and they go to write a paper or their thesis, I find they often will interpret that experiment in the old paradigm, the old idea. And I will say to them, but your experiment doesn't fit the old idea. Look, you've shown something that's inconsistent with that. They'll say, yes. So I say, point that out. Now you have to invent a new paradigm, a new idea. So I think we have to be alert to that. Well, that's a long-winded way of trying to answer that question.
And a question. And the comment concerns what David mentioned, and the importance of multidisciplinary or interdisciplinary research. And I think a lot of new findings come out when people from different disciplines meet together and then kind of, discuss, and then we form a team, and then we make discoveries.
Yes.
And I want to mention that at UCLA, we have a number of centers or organizations that promote multidisciplinary research. For example, there's Molecular Biology Institute, and all the molecular biologists at UCLA meet every week and discuss their topic or their science, and we eat lunch together. And that kind of generates a lot of new ideas and new research. We have Cancer Center, Johnson Cancer Center, and a lot of people are in the Cancer Center discussing about cancer research together with clinicians. And the third one is California Nanosystems Institute, and that's where a lot of engineers, medical people, physical science people, biologists, everyone get together and discuss. And these things promote new ideas and new discoveries, and that's extremely important. And that is the reason that we thought we should invite Joe Liu to this meeting. And that's because Joe Liu is the director of the state-of-the-art mass spectrometry. And that's a tool that's becoming extremely important in biological research. And so I'm looking forward to his talk tomorrow. And that's one of the things I do collaborate with him, and we get all sorts of new discoveries made. So that's important. Now, that's the comment. The question is, came up twice. There was a mention of laboratory of natural products and medicinal chemistry, and could someone kind of expand what this lab is about? That would be great. Thanks.
Yeah, now I can explain because in the Faculty of Chemistry, the natural product is the big group, strongest group in our faculty. 5 years before, we just focused the isolation and elucidation structure of natural compounds from medicinal plants, just like that. But now we focus in the isolation and identification active natural products following the bioassay. Yes, but we in our faculty we just we can test some simple activity such as antioxidant, anti-enzyme, anti-inflammatory. Other we must send to molecular biology department for testing some cytotoxicity, cancer cell lines, just like that. And now we have some collaboration with Japan, Korea for the other experiment in cancer research. Actually, in the medicinal chemistry lab, we just focus in natural product only. We not yet set up the medicinal chemistry research. Just do simple experiment. So in our faculty, we would like to have a collaborate with CMC Center for further biological activity testing. Yes, thank you.
Okay, I have small idea.
And our target is Alzheimer's disease, and now we start to do something related with breast cancer, and by doing visual screening, we have some potential compound for those diseases. So we look forward to collaboration on checking activity of our hits, and first so we did in silicon. So next step to do in vitro. So I look forward to collaboration with scientists from Ho Chi Minh City, and probably next step to do in vivo. And I'm going to talk about this tomorrow in more detail, but just to make some advancement.
So look like I'm allowed the last 2 minutes, so I will be quick. I think I heard some discussion about research on herbal medicine in Vietnam, which is really an important point in the way that that's the advantage the country has. We have a very vast resource of natural product. And actually, over the past years in our observation, there are already the effort in Vietnam to study the active ingredients as well as combination with the international community to apply molecular biology. However, the outcome hasn't been encouraging as we like it to be for many reasons. But one of that, I believe that because both sides haven't really combined their advantage into the collaboration of the effort to do so. I think I know one of the group where they try to use chip technologies and receptor drug signaling to screen for molecules, inhibitors. However, that is not the strength part of Vietnam technology, at least at this point. We hope after the collaboration with you, we will further develop the molecular screening technology. But I think what we do have now with the evolution of stem cell research. As you know, stem cell now has been a target for not only for therapy, but also for drug screening. We have the capability to transform any cell type, cord blood, peripheral blood cell, BM, bone marrow cell into some target cell, like the heart for screening herbal product for the heart, or even the liver screening herbal product. And the technology developed very fast. So that is the target that we think we can have. I hope that information will give you some idea what kind of combination that we can work with you to effectively develop a herbal screening in Vietnam. Thank you very much.
In our department. So in fact, I was a professor at Tufts University in Boston, Massachusetts. And 5 years ago, I came back to Vietnam and established the biomedical engineering department. So the reason for that is because I see that there is a great need in Vietnam about the medical devices. So therefore, our basic activities is about the development of medical devices for telemedicine. That means the patient can use those devices at home, and the information can be sent to a server, and the doctor can diagnose and take care from distance. And another activity is we use the nanotechnology to in the cancer research, and most importantly, using the ferric oxide to carry medication and bring that to the cancer site to kill the cells. This afternoon, unfortunately, I had another meeting, but one of my colleagues will greet you over there. So he's an expert in this field, so he may raise the interest from your group about that. One other thing also is when I was in the States, at one point I asked a director of NSF to be sponsor to develop the biomedical engineering in Vietnam. And his answer is, oh, Vietnam must develop other things than biomedical engineering, because biomedical engineering is a very advanced field, so Vietnam is not capable to do that. I insisted, and finally he agreed to sponsor a group to come to Vietnam. That was in 2004 to make a fact-finding trip to see if Vietnam had any potential about doing research in biomedical engineering. That's why I'm very glad to see that today, probably you do not give a hard time to the Vietnamese group to create this collaboration. Secondly, NSF afterward, they sponsor for us to organize the biomedical engineering conference. It was in 2005, and it was the first international conference in biomedical engineering. And after that, every other year, we organize that conference. And the last one was in 2012. And the next one will be in June 2014. I would like to extend our invitation to your group. If you can participate in this conference, that will be a great honor to us. Of course, you know, now we are in March, and to come back to Vietnam in June may be difficult for you, but it will be a great honor if you can accept our invitation.